All right, so welcome everyone. I don't know how it works. I mean, I'm happy if you ask as many questions as you want. I don't know if there's a format, but I mean, feel free to interrupt me at any time to ask any questions. So I'm gonna mainly sort of talk about two big concepts in collective dynamics of cells from a theoretical angle, and in both I'll start. I mean, most of the class will be sort of slightly historical perspective, and I'll go a bit on the blackboard to show some equations to derive some of the classic models of the field. And then if I have time, I'll show you in each case like one short example from some research that we've done in the past. So why are we, so let's define the terms, right? Sort of active matter models, I think you've sort of encountered already in a summer school. What are we talking about when we talk about collective cell dynamics? So I'm going to talk about sort of, I'm going to give you a few examples from biology, very simple experiments that show you that sort of, if you think about biological cells, they don't behave on their own. They don't sort of, it's not some diffusive process where everyone is uncoordinated and you give rise to sort of essentially sort of large-scale diffusion, but you have really interesting sort of collective dynamics that show up in really, really simple biological systems. And I think one of the simplest thing is like a beautiful work, it's quite old now, it's work in the 60s, which has been sort of really been incredibly influential in this idea of collective dynamics and the idea of chemotaxis, what impacts what cells do. This is like a ridiculously simple experiment. You take essentially E. coli, some bacteria, and you seed them on a 2D plate, and so you seed essentially a drop of bacteria on your job. And people know that everything depends on their motility. So they actively move, they have some flagella. But if every bacteria would just be uncoordinated and move sort of randomly, you would just get a diffusive front, right? You have sort of a heaviside function and sort of a drop that you put in the middle, and you would just get diffusion. Instead, what you see, and essentially sort of the white spots here are essentially proportional to density. So what you're seeing here is density profiles where sort of white is dense bacteria and black is essentially sort of the dish. What you see is that there's really interesting collective dynamics where the thing expands, the thing doesn't diffuse, it sort of expands radially as waves. So if you would look at a movie, it would have sort of a wave of a pretty well-defined velocity. And then it's not just one, it's that you have multiple sort of waves coming in the back. You can even sort of make them collide and they sort of interact. If you do two foci like this, your two waves interact and things. And this is really the hallmark that you have some complex sort of collective dynamics that happen in there. And again, there's really nothing special in here. This is just bacteria plated on a 2D plate. There's nothing that attracts them to the outside. This is just, they're plated on a uniform substrate. You can think about it as like on an infinite 2D surface. And so this tells you already that really, really simple systems have pretty interesting dynamics. So this is just, and this is a paper from the 60s. Then people played a lot with the different type of conditions. You know, you can play with the type of bacteria, you can play with the type of nutrient that you put. And so 30 years later, for instance, they systematized this a little bit, and they showed that if you do exactly the same experiment, again, sort of putting a drop of bacteria in the center and varying conditions, you can get sort of incredibly beautiful type of patterns, not just sort of radially outgoing sort of waves, but you can get sort of, you know, I mean, you always get an outgoing, a radial sort of outward going wave, but then in the back you get all of these beautiful sort of patterns. I mean, there's clearly a wavelength here. I mean, there's clearly a periodic wavelength here, but you can see that there's also like interesting sort of higher order structure where here it essentially seems to break. I mean, here you seem to have forgotten about, you know, the direction of the invasion. Here it seems quite clear that you broke symmetry between the direction perpendicular to the invasion front and the one parallel. I mean, there's just a lot of beautiful complexity in this.
Do you know what was done to go from the previous stage to this one?
So this is I think here mainly they play with the type of nutrient that they put on the plate. So it's they play a little bit with the type of bacteria. So I think in that paper I think they have multiple types of bacteria, and I think the main thing that they plate is the nutrient. So essentially the condition. But again, everything is always uniform. Yeah.
What's the scale bar? How big is this?
Sorry?
How big is this? What's the scale bar?
I mean, this is a petri dish, so
10 centimeters or so.
Something like this. So it's macroscopic, right? That's interesting. There's really something you really it's actually, yes, it's useful to have scales in mind. I mean, this is not an individual bacteria, right? This is sort of thousands of bacteria clumped together. Okay. So that's one example that you could say, okay, that's a bit in vitro, that's a bit artificial, you know, like does this happen? There's a system which has fascinated sort of mathematical biologists and physicists for quite a while. It's called, you might have seen it before, it's called Dictyostelium. It's essentially a slime mold, and it's a super cool organism. It's something that can transit from single cells to collective, basically. So it can exist as single cells. The cells can exist on their own, and they're happy and everything. But then when they have a stress, they tend to come together and form organisms. So it's something which is sits at a bit in between single cell organisms and sort of multicellular organisms, and can transit from one to the next. And so here, this is a movie I really like. You're not gonna, right now you don't see the cells. Let me just play the movie and you'll understand why I'm showing you this. So here again, you sort of see the cell. A cell is like almost a pixel here. For a while, everything is uniform. There's essentially cells everywhere. And then they want to aggregate because there's a stress. And so what you can see is that there's like global aggregation pattern, right? Where suddenly like everything, so every cell starts migrating towards a few foci. It's not perfect at sort of, you could call that phase separation, but it's not perfect at phase separating. So it forms multiple sort of a few foci, right? So these are like, again, thousands of cells that have aggregated. And then after it's finished, you know, it makes a slug, and the slug can sort of move around in the dish and sort of feeds itself as a multicellular organism. And this is completely reversible. You can also change back the condition, and you go back as single cells. So this is, again, a slightly crazy example where these cells are really efficient at collectively finding each other. You know, you could see that there were domains. If you go back to here, for instance, if you go back to here, which is a bit the critical stage of this phase separation process, you could see that there's multiple domains, right? There's clearly a length scale associated to this, where essentially, you know, up until the top third, you know, everything's going to converge upwards. And if you see the things on the top and the right, they're going to converge. They're going to make a different organism and a different foci, you know, migrating towards the top. So there's clearly some collective dynamics, some length scale, and some surprising capacity for cells to find each other, right? So some of the questions that I want to explore here is, how do they find each other? How do they collectively move? And can we get insights from very simple active matter inspired models to understand these kinds of things? So for starters, how do they find each other? I think it's quite clear that to migrate, I mean, I think it's quite clear, but I just want to hammer this point. This is not random diffusion, right? It's not something where, you know, they would randomly move and stick to each other. You know, you could think about something like diffusion-mediated aggregation or something like this. I think it's pretty clear that it's not that, right? There's like clear advection where they move over very large distances. It's not just that you randomly, you know, move and then get stuck on an aggregate when you find each other. And so if cells need to move over very large distances, over distances many times their own distance, it means that they need to actively migrate. And this is a very classical process in biology. This is the idea that cells can exert, I mean, everything that I've shown you here happens on a 2D substrate. And so cells can just sort of actively crawl, they can actively migrate, they can sort of exert force on the substrates. And so it's very well known that cells are able to sort of move in a directed manner and in a persistent direction. But of course, so now once you say, okay, cells are able to directionally move, they have a polarity and they move in the direction of the polarity. The questions from an active matter perspective is, okay, this sounds like some polar, you can describe this with some polar active matter kind of ideas, but the key problem that you have is you're going to have to find an equation for the polarity, right? In layman's terms, what orients cells? You know, what's the Q that orients the polarity of all the cells, and how how do you self-organize all of these polarities to go into one direction? Just to give you again one example. In a different system where people have this question quite a bit. This is again a minimal system, even closer to us. This is actually epithelial cells. So these are cells. This is a classical cell type that people use in the field of mechanobiology. This is called MDCK. These are kidney cells from a dog from the 50s. It sounds very artificial, but it's what sort of everyone uses as a model for cell migration. This experiment is a really minimal one. So I think the first people to do it was the group of Pascal Silberzan in a Curie Institute. essentially what they do is, again, so these are cells plated on a 2D substrate, and they simulate a wound healing, meaning that what they have is essentially there's a half plane that was occupied by a stencil, and what they do is at T0, they lift the stencil, and then suddenly you have essentially half a plane that's filled with cells and half a plane that's empty. And so essentially what you made is you made a wound without any of the inflammation, the scratch, any of the sort of actual wound sort of inflammation response. You essentially have like a wound in a dish where there's nothing that tells the cells that there's empty space except sort of physical, in a way sort of physical constraints, right? And so what sort of the group of many, what sort of the work of many people, of many groups in the past few years have shown is that these cells here are surprisingly efficient at closing a wound, right? So if you run the movie, basically, these cells again sort of always migrates towards the wound, and they're pretty efficient. That's again reliant on directional migration, so they need to actively migrate for this. Proliferation plays a bit of a role, but it's actually not the dominant role. You can block proliferation, and these cells are still efficient at closing wounds. So this is again something where active migration, active crawling plays a key role. And one of the surprising features from, and again, you can really ask the question, how do these, who knows, how do they know to migrate towards the left, right? What orients their polarity towards the left? For a while, one of the ideas in the field was that, well, the cells at the leading edge, they're special, right? The cells at the leading edge, it's very clear that they see a different mechanical environment left and right, right? They were called, so there's a big idea in the field for many, many years that these are leader cells. They're super migratory, and they essentially pull everyone, towards the left. But what the group of Shafit-Repa showed sort of 15 years ago is that if you actually measure mechanical stresses in this monolayer, if you actually measure the stresses that these cells exert, it's not the case. Actually, it's not just the leader cells, the cells at the edge. It's essentially every cell in the back contributing a bit, and so the cells very far in the back know that they should still go towards the left and contribute. So it's again an example of collective migration dynamics. And it's actually from one of our papers with the group of Tsuyoshi Hirashima. What he could do is to actually visualize in the system cell polarity. So the colors are not too important. Essentially, the green is the cell nucleus, and the red, again, it's the Golgi. It doesn't really matter. The important thing is that it gives you a readout of cell polarity. Essentially, it's known that cells exert their active polarity essentially in a vector that goes from the green to the red. And so what's cool with this is that you can draw vectors for each cell, by drawing essentially a vector from red to green. And what you can see is that it's not just these ones that are polarized, but cells sort of millimeters away from the wound are on average quite biased towards the left. And so this again begs a bit of a question on, information propagation. How do these cells know that they should go that they should be oriented towards the left, right? And just if you just recast this in a language of sort of in a language that might be familiar to statistical physicists, it's surprising because it's a 2D system and it has a long range order, right? It's essentially you have a polar 2D system that seems to show order over sort of distances of millimeters, which is always a little bit surprising. Okay, so that's a lot of examples. That's a lot of examples of the type of biological questions that are interesting and sort of and give you some hints, I think, of why it's interesting from a physics perspective. So essentially, now what I want to give you in this talk is like two big ideas from the field of how this long-range order can emerge and how can you make alignments. So one, and actually the two are going to come from slightly different communities. So the first one I'm going to talk about is actually something that if most of you are physicists, it might be a bit less familiar because it comes a little bit more from applied math and mathematical biology. The second one might be a bit more familiar because it really comes from statistical physics. The one that comes more from applied math and mathematical biology is the idea that cells are guided by diffusible gradients. Essentially, the classical idea, if you open a textbook on cell migration, what you would get is that essentially what happens is that in cancer, in development, in sort of, bacteria, in bacteria, essentially, they are in many cases sort of gradients of a chemical species that is essentially a chemoattractant. And as its name chemoattractant implies, it essentially attracts cells. So what cells are able to do is they're able to measure concentrations at their front and at the back. They're able to measure differences, and they're able to align and to start moving generally up the gradient. So usually sort of most diffusible molecules are chemoattractants, meaning that cells are able to migrate up in the direction of high high concentration. So this is again the classical example. So the classical example is that somehow something laid out a gradient, and then the cells read that gradient that was magically laid out from the start. This is the oldest idea. This is over 100 years old. The field of chemotaxis. Nowadays, there's been a lot more cues that cells can actually sense. There's a lot. So people have realized that there's a lot of other gradients that cells can sense. So one which sort of physicists like, for instance, is that it's shown, for instance, that cells can sense also gradients of stiffness. So if you plate cells on substrates that have gradients of mechanical of stiffness, they're able to migrate up the gradient. They can also migrate up gradients of friction. They can migrate up gradients of linkers. There's a lot of, there's essentially a lot of physicochemical cues that when they're graded, cells are able to migrate in response to this. So that gives you the cue already. So if you start thinking about equations now, if I write a polarity equation for my cells, my polarity is a vector. I want to couple it to other vectors. Well, if I have a chemical field and I can go up gradients of chemical field, the gradients of my chemical field or my mechanical field or whatever, that's a vector. So I'm going to be able to start writing a theory where I couple polarity to other things, right? So that's one of the ideas. It might sound satisfying, but again, the problem with it, the reason why it is not very satisfying, and essentially in all of these examples I've shown you before, essentially almost all of the examples I've shown you before were in vitro, where cells had been placed on a 2D dish. So there was no magical sort of gradients being laid out, right? There was no pre-patterned cues that any cells could sense. And so in a way, the question, it's a bit of a, we've only displaced the problem. If we say, okay, cells migrate sort of up gradients, you need to explain how the gradients come from in most situations, right? And so the type of model I want to talk about essentially are relatively simple. They consist of two equations. One is essentially tracking cell velocities and cell densities. So it's essentially a conservation equation for the cells. And then I also need to write an equation for how my chemical field is going to evolve. And the idea is if I couple these two equations in the right way, if cells can sense the gradient, but also shape the gradient, but also sort of influence the gradient in some way, I'm going to start getting interesting patterns. And that's one of the core ideas that I want to explore a bit that I'm going to explore a bit on the blackboard. There's just so essentially the core idea I'm gonna sort of try to demonstrate, and it's something really that has emerged over the last 10 years. Is it's not just that cells sense the gradient, so it's not a unidirectional relationship between, okay, you have a gradient and the cells migrate up. It's also that the gradients themselves are reshaped by cells. Essentially, cells can either eat up this chemoattractant molecule or they can degrade it, and therefore there's a feedback where cells can sort of reshape their own gradients, and that has the potential to give rise to interesting dynamics. And just the last example the last example of this, because it's a beautiful paper, before sort of going on the blackboard, is a paper that drew quite a lot of attention by Rob Ensor a few years ago. Essentially what they took is they took cancer cells, and they showed that these cancer cells could solve mazes. So the idea essentially is that it's a maze made of 2D macrofluidics, and then you flush it with chemoattractants, but the chemoattractants is uniform. So initially you have a field of cells, you have a field, and you have a field of chemoattractant, and it's completely uniform, and then you put the cells on the left side. And the question is, there's the maze, and the question is, can the cancer cells find the maze at the end, or are they going to get lost? How efficient are they at solving the maze? And I think it's quite clear that this is important, right? Because if you think about cancer cell metastasis, in vivo, it's a pretty complex environment. They can get lost in a lot of different places. So the idea of cancer cells being able to guide themselves in a given place is clearly something that is sort of pretty interesting, I think. And what is beautiful about this paper, and something which is quite incredible, is again, there's no gradient, so the chemoattractant was flushed completely uniformly in a system. But if you look at these cells, if you look at the cells that are coming in, they basically make extremely few mistakes. There's a few that get lost in the dead ends, but they almost instantly recognize that you should go towards the dead end. And again, this is an example of collective dynamics because they can sort of they can sort of see things that are much larger than their own size, and they're pretty efficient actually at solving mazes. And again, without sort of spoiling too much of what I'm going to talk about before, the conclusion of this paper is something exactly what I've sort of alluded to just before. It's the idea of self-adjudicated gradients. The idea basically is that initially the chemoattractant field is uniform, but you do break the symmetry by putting the cells on the left. So if you put the cells on the left, they start eating their own attractant. So they start eating the chemoattractant, and they're going to make a gradient, right? Because if they eat it on the left and it's everywhere, that starts forming a gradient. But that gradient, because you have a source of chemoattractant here, that gradient is diffusible and is sensitive to boundary conditions. So essentially, the gradient is going to be different. The gradient that's going to form towards the bottom is going to be different than the gradient that's going to form towards the top. And essentially what happens is you're eating up a diffusible field, and your diffusion equation is essentially solving the maze for you, because it's essentially telling you what's the path towards the reservoir. And so I really like, they've done quite a bit of modeling actually in this thing, so they actually solve the diffusion equation on these mazes and everything. It's pretty convincing if you want to have a look. But it's just, again, a beautiful example where you have collective dynamics emerging from essentially pretty simple thing, right? You have one type of chemoattractant, one type of cell, they eat it up, and you can give rise to interesting things. Okay. Yeah.
So, is there any connection between diffusion-phoresis and chemotaxis? So, diffusion-phoresis is the motion of a particle with a velocity in a gradient of a solid.
Yeah.
So it's a purely passive, osmotic, if you want it. And here is, of course, driven by V.
Yeah.
But if you had a way to kill the cells in the same gradient, and they, I mean, this is a bit of a difficult example.
No. I think I would think the effect of diffusion phoresis are extremely small, because these gradients are pretty shallow actually. So I mean, there's a whole field on information bound on how much cells can sense. I mean, it seems like quite a few cells are able to sense sort of differences at the back and front of orders of a few molecules. So they're really hypersensitive actually. And so in almost all of these experiments, I think, for instance, when they kill the flagella or when they kill ATP, the cells basically don't move actually. So it's really an active. I think it's really an active process. Yeah. But that's an interesting idea. Yeah, by the way, I'm going to go to the blackboard, so any questions you have on this part of the talk, please ask.
Sorry, but also when you remove the film and the cell invade the empty environment, in that case, we haven't chemotaxis, or yes, in the region of
sorry,
the empty.
You mean in this example?
Yes, in this example, I thought that the empty region was empty, so without any chemotaxis or something like this. So I don't understand.
So that's a good point. I hope I'll get to that. So I mean, but I don't think I will get to it actually. So we worked on this. Of course, you could imagine as chemotaxis. I mean, here you don't put any obvious chemoattractant. I mean, you put nutrients or things like this, so I guess there could be some things where the cells sense their own nutrients or chemotaxis. We don't think it's the case, because I think also people have applied sort of microfluidics shear on it, and I think it's so I don't think it's actually chemotaxis on this. So we have a paper on this from a few years ago actually. So what we think is going on is we think there's essentially waves. So there's mechanochemical waves happening in this. So basically, if you look at density or different sort of signaling molecules, there's essentially waves that go from the back to the front. And so what we think, so it's a paper Bucock et al. From 2020. And so what we think is that there's essentially sort of mechanical waves going from the edge to the back, and this is what orients. This gives some information to the cells. So they're essentially active waves, so they propagate infinitely far in the bulk, and we think that that's what gives the information. So I think we think in this system it's actually a very different mechanism for polar order.
Time when every layer of cells change the alignment.
Yeah. I mean, at some point, I mean, early on, yes. So you see development of. I mean, the order gets more and more perfect in time. So initially it's mainly these ones that are ordered, and then if you track sort of the polar order, essentially as a function of time, you get essentially a diffusion of the polar order, so it invades essentially the whole thing. So there is some special intertemporal information which you've also modeled if you're interested in.
Okay, thank you.
Did you perform this experiment without nutrients?
I mean, I guess nutrients tend to die, so I guess it's difficult to.
Some cells can withstand
Sorry?
half a day. Some cells can stay in quiescence.
Yeah, that's a good question. I don't know what. I actually don't know what will happen if you don't put any nutrients or anything. I actually don't
The
know.
cells in the middle are lower in oxygen and nutrients, and they always try to find better place for living. So I think the cause for my whole colony can move also can.
It could be. What we're not totally sure with this is I think I remember that for instance people did these experiments with microfluidics flows where you apply shear on different things. And I don't think it changes much actually. So that's one thing. The other thing is that in the previous experiment in Exocilium, they had to go in sort of microfluidics channel, which are 2D, where essentially there's very little bulk, right? Here, these experiments are done, on in a petri dish where there's like centimeters of media, and these cells are only 2D. So I also don't know how much, oxygen gradients would form. The things would sort of. I'm actually not sure in these kind of sort of 3D systems how much gradients would form. But that's a good question. I mean, essentially, I certainly do not want to say that all of these situations are. I mean. I'm going to give you sort of two kind of mechanisms that work on this. So I certainly don't want to say that sort of one mechanism applied to every single case in biology. That's for sure. So there's many open questions in the field certainly. Any other question on what I showed so far? Okay. Can I light up Can I just light up this because I'm going to go on the board? No. I think it's. Okay. Oh, sorry. All right. So very. I mean, I'm going to go a little bit briefly just on the kind of equations that we want to describe with this. Essentially, if you think about these kind of experiments I've shown you, so let's take the simplest thing, the first thing I've shown you, where you see these rings of bacteria, and you've seen that you make a drop of bacteria and it expands as a ring with a pretty well-defined velocity, essentially of a front. The simplest thing you can do is you say, okay, my ring consisted of thousands of bacteria, so I don't really need to track every individual bacteria, right? I can go to the continuum limit. So what I want to do is I don't really care about individual bacteria. The object I care about, right, is a field. Essentially, I feel about my concentration of cells, right? That's what I care about, really. What I care about is it's a continuum thing, and I want to care. What I care about is writing down a continuum theory on this, right? And so, if I want to write a continuum theory on this, the first thing I have to write is let's not take any assumptions of physics. Things need to be conserved, right? So the first thing you write when you write a continuum theory like this is I want to write a continuum equation, right? So what I want to write is I want to write something like this, where essentially this is growth and this is advection, right? This is the most generic equation I can write with this. Actually, I'll write it even more. I'll write it even more general. Let me write it as terms of flux. Here, so if I want to write, if I want to see the evolution of a field C of concentration of cells, my field at a given time and point, it can evolve through two ways. it can have a flux, so cells can move from x to x plus dx, right? And so now the big question is, what do I write for this flux here? And then, because these are cells, they can also grow and they can also sort of proliferate and die, right? So I have something which is a growth term, which is essentially a non-conservative term here, right? So that's the simplest thing I can write. And now what I need to write in my theory is two things. I need to write something for this, and I need to write something for this. So I'm actually going to go back, because I think it's a bit useful historically, so this is essentially the idea of using this kind of partial differential equations for collective dynamics is pretty old. And the simplest thing, and people have been looking essentially for collective solution, and one of the simplest collective solution you can find of this is a traveling wave. Sorry, yeah.
Why do you write a minus sign for the flux?
Sorry, I had an input, yes. It was minus rho VA, so obviously.
Thank you.
So the simplest thing we can look for as a collective dynamics in this is traveling wave solutions, right? One of the things that we're interested about is something that travels at a constant velocity, where a peak of cells sort of migrates outwards. In this, the oldest idea on this is one which is essentially Fisher KPP. So it's a pretty old model. Now it's a model that was essentially derived independently by two groups. It's from the 30s. So it's essentially a model that is thought for population dynamics. So independently worked on by Fisher on one side, and so the K here is the Kolmogorov that you know from turbulence, and also other people. And essentially, the idea of this model is super simple. You take this, and then you say something incredibly simple. You say that my flux here is minus d grain of C. So essentially that's fixed law, right? So I'm saying that my cells just completely randomly diffuse. So it actually goes against everything I've said you about active cell motility, but it's actually useful to think about what happens without cell motility. That's like the zeroth order term that can happen, right? So it's just assumes that every cell randomly moves. So this is basically fixed law, right? So very familiar. And then the other thing I can write is I can write that cells divide. So if I write that cells divide, I'm going to write that sort of they can exponentially grow, right? So I mean, this is something where I just have exponential growth of cells. That's not very physical, right? Because that's going to blow up. So I want my cells to grow, but I also want them to stop at the critical density. And so the simplest thing to do is to write the next term in the expansion, basically, is to say that there's something that's called a carrying capacity, where essentially sort of cells stop growing after a certain amount. And so what this tells you essentially is that when cells have a low density, C is close to 0, you grow at a rate K, and then cells stop growing when they're at a concentration of 1, right? So this is a really, really simple kind of model where you've made two super simple choices for your flux and for your growth, right? Okay, let me just put this up a little bit. And so if I combine it, if I combine these two things and I go in 1D now, what I have, right, is I have a Fisher KPP equation right, where I have just diffusion and growth. And typically the way it's solved is I'm solving as an initial condition. So that's my C x of t equals 0, where essentially I'm going to put a Heaviside function, right? This is the drop of bacteria I was putting. And the question is, what happens here, right? There's two effects. That's one effect, that's diffusion. So if you would have this term alone here, you would tend to, slowly diffuse in this direction. But then there's also an exponential term here that tells you that these, as soon as there's a little bit of cells here, they're going to grow like crazy, and so you're going to sort of move this up like crazy because you have exponential growth that goes extremely fast here. So I don't want to go into the details of. I don't want essentially to go in the details of these kind of equations. Essentially, what you want to do in essentially all of these examples is you want to look for a stationary. You want to look for traveling wave solutions, and so essentially you want to look for solutions where you look at some coordinates, which is x of mi minus t. Essentially, you want to say that my concentration field doesn't just depend on x and t, but if it's a traveling wave solution, it's going to depend on a single reduced coordinate, which is x of v minus x minus vt, which means that essentially, if I rescale by time, I'm just going to get the same thing over shifted by a velocity of v. And the reason that this is very convenient is because essentially this becomes minus v. If you take my time derivative, it's pretty close to my z derivative up to the speed v. So I get some dz of c here. Similarly here, so I can essentially even drop, and the prime is now a derivative with respect to z. I have c here, and then I have a k, c 1 minus c here. And here I'm already getting something quite manageable because now I reduce it from a PDE to an ODE, right? And essentially, what happens with this is that you can see, you can actually show a few things. First of all, what you can do, what is always very useful in these kind of things to solve for this is that you can see that this is nonlinear. If it wasn't nonlinear, you could solve it super easily, right? If I would sort of cancel out this term here, then it's just a linear. I mean, it's just a linear ODE, right? It just would have an exponential profile. But the thing is, anyways, I'm looking for traveling wave solutions of this kind, right? So if I look ahead, ahead of the front, x equals plus infinity, right? I always have c going to 0. So then I can always neglect my c here, and then I just have a first, and then I just have a linear ODE, which means that in ahead of the front, c is just going to be an exponential minus x of lambda, right? Because that's just the solution. That's the physical solution. That's the physical solution of this linear ODE here. And why is this useful? Well, this is useful because first of all, I've solved already sort of the front. I mean the front of the wave of my traveling wave here. You can do something very similar to solve even the back actually, because again in the back c is very close to 1, and so you can linearize up to here. And so if you piece all of these things together, what you see is that there's actually a lot of traveling of velocities that are forbidden, and essentially. The only velocity, I mean, essentially, the velocity that gets selected with this is d over k, d times k here, sorry, d times k here. And so essentially, it's a super simple model in which you get traveling wave solutions. And your traveling waves go at a very well-defined velocity, which is, I think, a really interesting compromise of diffusion, which would make you go as a square root of t, right? And k, which is your exponential growth, which would make you grow exponentially, essentially, right? So there's this kind of really interesting compromise on this collective dynamics, where this would drive you towards sort of towards diffusive thing. This would drive you towards exponential thing. But there's a compromise between the two, where essentially the cells diffuse. It makes them go into regions that are less dense. It allows them to proliferate. And so the solution essentially between them is to have a constant velocity. And so essentially what you do is you can explain through this why, if you start from a drop of population that just does diffusion and growth, you're going to have a constant velocity and sort of collective dynamics where you invade space in a collective manner. Yeah, sorry, that's a question.
Is it simple to show how the parameter lambda depends on t? Because I think it depends on the ratio.
Yeah, so you can show. I mean, you can solve. I mean, I don't want to spend too much into this. You can solve. I mean, this is a first order. I mean, this is a first order ESI, so you can show that your. I mean, you can sort of. If you plug.
It's just standard from solving the equation.
Exactly. You just solve this. You just solve the second degree polynomial, and actually that's where it comes from. That many waves are forbidden essentially, because if you look at the determinant, your determinant is going to be your lambda squared minus some v's, and so there's going to be a lot of v's in which the determinant is negative, and so you get imaginary values, and so they're forbidden. So that's where it comes. That's where a lot of different wave velocities being forbidden comes from exactly. Yeah.
So for c equal to 0 and c equal to 1, use better the equation, right? Because f is 0. So I would expect the traveling wave solution not to work very well close to zero. But work well in the region between.
But it's never exactly zero, because the thing is that I mean the traveling wave solution sort of links 1 and 0, right? So it always develops basically. I mean, it always develops as soon as you have the right initial condition. You always develop according to these conditions, right? Because you can't just set C to 1 and 0, you see what I mean? Because it's just itself a parameter.
Yes.
Yeah. Okay.
Quick question. Is there a kind of simple hand-waving intuitive picture? Why would I get, I mean, okay, dimensionalizes gives me d times k on square root. But is there a kind of simple intuitive picture why kind of on one hand I'm playing diffusion against essentially creation of, and why do I get the velocity out of this?
So, I mean, one thing you can say, I mean, the simplest hand-waving argument is just scaling, right? Is that if you know that you're getting a velocity, that's the, I mean, you know, this is a diffusion, this is sort of, you know, length scale, I mean, this is sort of L squared over t, and this is a 1 over t. So that's the only way to make a velocity. So that's that's a scaling, that's one scaling argument. There's nothing else in that. The intuition, I think for me that comes from this is again that to me it's not trivial. The intuition is that essentially you diffuse. I mean, no. So if you would have no diffusion whatsoever, if you have no diffusion whatsoever, and you would start from a perfectly heaviside thing, nothing would happen, right? Because you would not grow here and you would be above grow here. So what happens in this kind of limits is that you have the cells start to diffuse a little bit, and then they get amplified, and then sort of as soon as they essentially when they migrate. So I mean, I don't know, if you make a stochastic, you know, if you make a stochastic agent-made model where you can shift here to here, basically, you know, if you make essentially a jump equation, you know, these cells are equally likely to jump left and jump right, but when they jump right, they're more likely to duplicate, and so they get overreach, you know, they get overabundant in your stochastic process, right? So then you bias your diffusion equation, because essentially you get out of your diffusion equation because you enrich one step versus the other. You see what I mean? Yeah. Cool. Oh, thanks a lot. Okay, but this is actually not sort of the. I mean, again, this is just to show you that you can get sort of already traveling wave solutions in complex dynamics without any actually directional movement. But what I've just told you before is actually you do want to incorporate active migration in this picture here, right? So let me just erase this. And the question is, what's the simplest thing now I can change? What's the simplest thing I can change to this to add something to add advection? So the simplest thing I can do, right, is to say that now I just don't have diffusion, but I have advection as well. So I think it's quite clear, right, that if you have directional motion, what is going to show up is an advective term in a flux, right? That's essentially you have something that's proportional to c, and that's advected at a speed v. So that's the simplest addition you can give to this. And now what I want to talk about is so essentially what I'm going to go through is the Keller-Zegel model. And that's 1971. It built on some previous work of mathematical biology, but it's actually a really important and influential model in the field of applied math, which is essentially to model what happens if you had the advection here. And so what I'm going to do for now is I'm going to actually simplify to the maximum. In the simplest Keller-Zegel model, I'm actually now going to forget all about the growth of cells. So I'm going to go to the different limits where I'm going to put something interesting in my advection term, but I'm going to put literally nothing in my growth. And of course, you can combine the different models, and there's like infinite papers trying to combine the different things. But I'm going to show you now that you can also get interesting dynamics if you relax. Essentially, if you put sort of some velocity here and you put growth to 0 here. So let's just erase all of this. It doesn't erase very well. Hopefully. Okay, so I'm gonna remove this. So let's add my advection term here. What I hope is very clear from this is that first of all, the first thing you need, that when you added this thing here, is that you needed to add, you need to write an equation on V, right? The problem is that now you need to, now that I said that my cells have an actual velocity, so this is again cell velocity. So I actually added a variable this time, so I'm going to need an equation for it. I could sort of take an active matter approach and write a very generic equation for how V evolves and is coupled to my polarity and is coupled to the vectors. I actually don't want to do this because it would get a bit too complicated. The key thing is I want to say to give you the intuition that I gave you before, which is that my velocity of cells migrate up a gradient of chemical. So I want to say that V is just, I'm just going to say that V is an instantaneous function of a gradient of chemical N. So this is some chemoattractant N, and typical assumptions in the field, for instance, is to say that you're coupled, that you sense sort of absolute gradients, or the other possibility is that you sense relative gradients. You know, that you can sense that what I'm sensing is some not some absolute difference between the front and the back, but I sense some relative things. You know, there's ways of cells to sense different things. So that's okay. So now I coupled my V to a gradient to a gradient of concentration. Of course, yet I introduce it. Sorry, yes. Sorry.
I think I got lost between V that is a function of the gradient.
So V, I'm just saying V is just proportional to my gradient of chemical.
And then,
so sorry, it's two options. So this is option 1, and this is option 2. Sorry,
okay.
It's just two modeling choices that you can make. You can say that. Yeah, sorry, it's a bit unclear. I mean, there's different options in the literature. This is based basically on the biology you need to model. I mean, people have modeled the details of cell sensing or something, but this is essentially you know, coarse graining everything that happens inside the cell and all of the sort of sensing that happens and saying instantaneous, which is a very big assumption, but you know, let's make it because it's a bit easier. Of course, now I introduce yet another variable N, and what I've told you before is I don't want to impose N, I don't want to say that N is already a pre-patterned gradient because then things would get boring. What's interesting is when N is itself an evolvable variable, right? And so what I need to do now is I need to write a conservation equation on N. So what do I write? Well, I don't have many choices. My N is a diffusible molecule, so it diffuses. It doesn't really make sense to write an advection term, right? Because it's a passive species. It's advected. Sure, when it's inside the cell, it's advected, but it's minuscule. I mean,
you agree that this looks diffuse formally? Formally, I mean, I'm not saying that this is the same, but the description is the same as diffuse.
I mean, the difference is here. I mean, there is a difference, which is that I'm going to write the key term here. Is here, right? The key term in my equation is that my diffusible field depends on. Sorry. The key term here is I'm writing that my diffusible field, my field of chemical, depends on is an arbitrary function. You can say it's produced, it's degraded, but it also depends on the cell concentration. This is the idea that this chemoattractant can be either produced by cells, in the sense that there would be a positive term dependent on C, or it can be degraded by cells, saying that there would be, you know, for instance, that you would have a negative term dependent on C here. And that's actually what does what generates most of the dynamics here. So what's the simplest thing? What's the simplest thing I can write? Well, I can write this, for instance. Let's take, you know, the first example, and then the other thing I can do is I can take, so the original choice of the Keller-Zegel paper is saying simply this. What does this correspond to? This is saying that my growth term, my chemical essentially is only degraded, so it's not produced by anything. I put it initially, and then it's degraded, and then the degradation term is proportional both to the cells and the chemoattractant, and this makes sense, right? Because I mean, this sort of makes sense because essentially what happens is that you need it's like It's like chemistry, right? For the chemoattractant to degrade, you need one chemoattractant molecule in one cell. The cells eat it up, and so that's why you get a multiplicative term here, right? That's clear to everyone. So that's the absolute simplest thing you can do here. I mean, it's not actually in the actual Keller-Zegel paper. It's actually written like this, which is very weird because it means that the chemical field can go negative because it's which is a little bit. I mean, there's a bunch of mathematical problems with this, but I'm going to sort of use the slightly corrected one from subsequent papers where you have this, and because there's an N here, this ensures that the concentration field never gets negative. Anyways. So what's the idea of this? Again, we're not going to solve the equations. What's the idea behind this? Well, the idea behind this is if I start from a uniform chemoattractant field, nx of t equals 0, and then I have, again, a drop of cells. Now here, so, I have, sorry. So if I have some cells here that are like, a drop on the side, this is X, and I have a uniform field of chemoattractant, what happens later on, so if I look a bit later, let me use some color, what happens instantaneously is that here, there's some cells, so it's gonna start, so that field of chemoattractant, I should write it red, so what happens in time is that this, instantaneously what happens is that this field of chemoattractant is gonna be depleted, right? It's eaten up, it's eaten up here and it diffuses, so there's a diffusion, so there's a length scale that shows up, and essentially what you make is a self-generated gradient, right? You make, you deplete the chemoattractant in the regions where the cells are. But then what you did now is you made a gradient. So if you made a gradient, what happens? Well, the cells, these cells here, suddenly they sense something, right? Because they sense a gradient here. So if my chi is non-zero, here I have a gradient of n, so these cells start to move forward, right? So then if I look at a later on time, what happened is that my cells started migrating, and they started eating up the gradient. But this thing is going to keep going on, right? So the cells migrate up. As they migrate up, they eat up the gradient, but which makes a traveling gradient. Essentially, what they do is they make their own traveling gradients of molecules by eating it. And then because this thing is diffusible, there's always some gradient in front of them, and then they can keep on going essentially forever if you look later in time. So this is essentially what happens. And in fact, if you simulate these equations, this is exactly what happens. So essentially, you start with this, and there's a traveling wave solution, and there's a traveling wave solution that shows up, where essentially the core idea of this is that as you move, I keep reshaping my own gradient, and I keep making the signal that enables me to move in front of me. Is the idea clear for everyone? Yeah.
So very naive question. Yeah. Is the velocity getting down in time in this picture, or is it constant?
So the nature of this is a mess. The core idea, it essentially depends. Essentially, if you write it this way, the velocity is going to go down because there's no source of chemoattractant, so the chemoattractant is going to exhaust. So actually, I shouldn't draw it like this. I should draw like an exhaust. I mean, I should show an exhausting source of chemoattractant. There's again an infinite sort of applied math literature about if you write every polynomial, you can write any polynomial here and any type of things here, and there are some conditions where you get perfect traveling waves. What happens usually is that you have like quasi-traveling waves. Basically, what happens in our experience in many of these things, even if you don't have perfect traveling waves, what happens is that for a very long time you have a near constant velocity or a velocity that evolves over a very long time, where you can locally define it as a traveling wave. Basically, that depends. I mean, there's a bunch of math of very detailed theorems, on exactly how the nonlinearities control this. But it's pretty general that essentially, for all intents and purposes of doing an if you try to explain an experiment, you're not going to explain infinite times anyways. So for all intents and purposes, this picture of traveling wave is actually a useful one. Yeah. Any other question on this? No. Okay. You can actually derive. There's another very interesting, I think. Picture. There's another very interesting limit for this, instead of assuming this as a loss, is if you remember the second slide of the talk, if you looked at this bacteria, right, there was one solution which was traveling wave, right, where you had like a band of bacteria that was expanding, which looks very much like this. But then if you looked at the next slide, you know, there were all these foci, right, these patterns where you had this periodic pattern formation that was forming. Yes?
So at the beginning, this traveling wave was a band instead of a star. So does this drop on the backside of the wave?
Yes, again, so that's an excellent question. Yes, it does. Again, that's related to Roberto's question. Essentially, when you don't have any new chemoattractant showing up, it happens very generically. The general idea is that if you make, for instance, your heavy side, if you make, let's say, it also depends on the exact parameter, but essentially the intuition is if you make your initial condition, your heavy side, you know, your initial job too large, essentially compared to the length scale of diffusion, then what happens is that there's only one part of the cells that start migrating, and then you have a pulse, and then in the back it doesn't happen, and because you don't replenish your chemoattractant, essentially there's nothing that sort of takes away the cells in the back. And so again, depending on the nonlinearities that you use, depending on whether you replenish your chemoattractant or not, you can get traveling pulses or traveling waves. It actually depends. We have a paper we just put on the archive also where we looked at this in different cases. It also depends on the sensing actually. One thing which you can see here is that this is way more sensitive, right? Because here, even if you have very few chemoattractant, you can still be super sensitive. And so in the back of the wave, this is much better. So it depends a little bit on the nonlinearities of the problem, but you can get both. That's a very good question. All right. But then the other thing I wanted to talk about is, if you remember, there was also periodic pattern formation, right? So I've shown you these bacteria. There were these periodic dots which clearly cannot be explained from this because here there's a traveling wave, but there's no patterning. There's no wavelength of patterning. But the cool thing of the scalar-Zegel model, it says it's an incredibly versatile one, and you can make an incredibly simple change and get some completely different phenomenology. And what people realized quite early on is if I take my function g to not be minus c plus n, but instead I say that sort of this is c minus n. So why am I saying, what does this correspond to when I say that? Well, now what I'm saying is that my chemoattractant spontaneously degrades, anything degrades, right? But what I'm saying now is that the cells produce the chemoattractants, right? This is a positive term where the cells produce the attractants. And this completely changes everything because now I'm not eating my own chemoattractant. I'm essentially sort of secreting it. And so essentially a change of sign. And so why am I telling you this in the context of pattern formation? Well, the idea is completely different. Right now, you're going to make essentially the reverse. If you imagine that I concentrate a bit locally, if I imagine that I concentrate a little bit, my cells here, just get this. If I have a little bit of if suddenly I concentrate a little bit my cells here, my cells produce the chemoattractant, so I'm going to make a chemoattractant here, and then I'm going to reinforce it, right? So this is something that now, instead of tending to spread cells where the cells move away from their regions of highest density, is the reverse. It's a self-attracting state where essentially the cells want to go towards regions of high densities because the regions of high cell densities are the regions of high chemoattractant concentration. So now it's actually the reverse where you're going to make pattern formation. And again, I don't want to actually derive these things exactly, but you can actually start from a uniform state. So the classical way you can understand this is essentially doing a perturbative theory, where let's say that my concentration. Let's say now that I started not from the heaviside function, but I started from a uniform field of cells and chemoattractant. I can do a linear stability analysis where I say, okay, let's look at how small perturbations. Let's look at how small perturbations of cells and chemoattractants are they stable or are they not stable? So I hope you're familiar with this. Essentially, what you're doing with this is that the idea of a linear stability analysis, right, is that you try out different functions, right? So you say that delta C is going to be delta C0 exponential of IQX plus omega t, right? So essentially what I'm saying is I'm going to try out a bunch of patterns, a bunch of periodic patterns, and I'm going to see whether a specific wavelength grows or shrinks. And essentially, if I plug this, I mean, it's again relatively simple. It's essentially relatively simple to do, right? If you plug these two things in these equations, you're going to essentially have a second-order polynomial equation on your omega. And so if you look at omega as a function of q, so omega is the growth rate of my perturbation, and q is the wavelength. What I'm very generically going to have is I'm going to have an instability that looks like this, where here this is the region that's unstable, and the region that's stable. So essentially, what this tells you is that essentially the large q, I mean the large wavelengths are stable, the small wavelengths are stable, but intermediary wavelength becomes unstable. Essentially, why is it intermediary length scale? Well, it is intermediary length scale because it's the length scale at which essentially you can build up this diffusion, this diffusion of your chemoattractant. And essentially, if you look at your criteria for instability, again, you can solve this. I'm not going to. I mean, you can solve it at home. It's relatively simple. Essentially, your instability criteria in this equation is something which is quite interesting, which is you need to be for it to be unstable. You essentially need chi over d. What are these chi and what are the d's? Let me remind you. Chi is my sort of chemotactic sensibility. It's my ability to migrate up gradients, and d is my diffusion. And if you think about it, it sort of makes sense. Because, you know, if your diffusion is very large, diffusion opposes patterns, right? Whereas the driver of my instability, again, is this chemotactic sensibility, is the fact that I tend to sort of move towards regions that are high in chemoattractant. And the core idea essentially is that these are essentially anti-diffusion process. You know, what happens that if you think about most physics process, what happens is, you know, classical diffusion, where, you know, if you have a local concentration, if you have a local peak of concentration, cells or particles tend to move away, to diffuse away from the peak of from the high concentration region. What happens in these kind of models is that the regions of high concentration of cells are also regions of high concentration of chemokine, which means that regions of high concentrations attract more attractive. So there's a flux. So essentially what happens is that there's a flux from regions of low concentrations to regions of high concentration, which is essentially negative diffusion. So essentially in all of these instabilities, these are advective instabilities where essentially you have essentially a negative diffusion term, effectively a negative diffusion term that opposes a positive diffusion term, and negative diffusion is unstable, right? I think that's quite intuitive. If you put your diffusion coefficient to a negative value, that's something that's unstable. And so that's the core of this, that's really the core of these instabilities essentially, is that you, yeah.
Look into dimensional analysis, it looks to me like you're missing some kind of length scale, because chi, from your definition, looks like it's 1 over time. It's a frequency, right?
Yeah.
And d has lengths in there.
Yes. So I non-dimensionalized. So I did. I mean, there's some dimensionalized thing that show up. You can see that this. You can see that here, for instance, should have some
prefactors, right?
Should have some rate constant exactly. Yeah. So these would show up in.
Okay. But that length scale is not important, right?
No, I mean no. This is the key thing is this.
Okay. Thanks.
Okay. Cool. So is there any question? So is there any question on this? So this was just the example I wanted to tell you, that if you essentially to just recap this part of the thing, is that you have pretty rich collective dynamics just by coupling cells to a diffusible field that you can have very easily, essentially traveling waves where you sort of explore empty regions with the constant velocities as soon as you degrade your own attractant. And you can have pattern formation with a periodic length scale that shows up as soon as you produce your own chemoattractant. So that's a very, very simple way that you can essentially have something that looks like long-range order. You can have sort of essentially highly correlated velocities of cells. And it doesn't really come from mechanical, from a mechanical picture. It comes from sort of, you know, coupling things to to a diffusible field. So yeah, I'm much longer than I thought, so I'm not sure I will have time for the second part of the talk. I will maybe go back just to give you sort of some flavor of how this is useful to and apply to data. Yeah, sorry.
Just a curiosity, maybe you have already said, but what happened when the diffusive flux and the chemotaxis flux are the same and the opposite? So we are in a kind of stationary state. Something of strange happen, or just nothing?
I mean, not much would happen. I mean, it's a linear instability. I mean, it's a linear instability. So if you just at the threshold, you're going to sort of, I mean, you might have some. I mean, then fluctuations are going to play a role, I guess, right? There's. Okay. So I would think that if you actually simulate it, you're going to see. I mean, because this. This instability really shows up instantly. So if you look at the threshold of instability, it's really a finite wavelength thing. So I would think that if you're just at the threshold, your fluctuations still are going to have that correlation length.
Okay.
Yes, I'm not going to do the second part of the talk. I'm going to sort of I'm going to use the last sort of 15 minutes to give you actually an example of some really recent paper that we've put on arXiv literally last month, where we actually use this concept and generalize them. So I think it's useful to. How do I turn this back on here? Right? How is it on? This one? Oh. So, so this is so I just want to show you a little bit of an actual application to actual biological data of these kind of ideas. So this is just a summary, right? Cells can self-generate gradients by consuming their own chemoattractant, and there's a bunch of references here of papers where people have shown. So again, there's a huge mathematical biology literature on this. These are, for example of biological papers where this has been found in a number of developmental cases, cancer cells. So there's quite a few examples where this shows up. This last paper, this is something we worked on with an experimental group doing immune cell migration, so the group of Michael Six in IST Austria, and this was applying it to immune cells. And I think immune cell is a particularly interesting example because if you think about immune cells, if you have a wound, let's say here, for instance, your immune cells in a lymph node, they have to get there, but these are huge distances when you think of them, right? When you think about it. Even if you have an inflammation signal, you need to make, these cells need to be attracted by it, and so you need to form gradients over the length scale of a meter, and people have recognized that if you put orders of magnitude into this, essentially there's no way you can sense a grain over, you can make a grain over a meter that cells are gonna sense. It's just too shallow. So there's been an idea in the field that, that self-generated gradients are better because self-generated gradients, it's not, you don't need to make a gradient over 1 meter, you make a local gradient that travels with you, right? Which is sort of much more sensitive as something. So we showed this in this paper that an immune cell type was able to self-generate their own gradients. But one thing that's interesting about immune cell migration is it's not just one cell type, right? To do immune cells, the immune cell response, you need lots of different cell types that come together, right? And all of them have different roles, and I'm not an immunologist, but it's really important that different immune cell types interact with each other. And in most of these theories, actually, people always consider like one cell type interacting with a chemoattractant. But here, what we wanted to ask is, can different cell types communicate to each other? Can we write a multi-component case where different cells are asymmetric and interact in a non-reciprocal way? So for instance, one idea that we had is that if you think about multiple cell types that have to go towards this, maybe not all of them have to generate the gradients, right? Maybe one cell type can generate the gradient and the other can surf on it, right? Because that's a slightly more economical sort of perspective. And in fact, so this is the PhD project that was taken up by Zana, and this is the work of a postdoc in my lab that's just left to start his group in Sheffield. If anyone's interested, is recruiting students or postdocs. And so what we've shown in the earliest paper that Mehmet was also involved in is that one immune cell, or dendritic cells, they can self-generate their own gradients. So it's exactly what I've shown you before. They eat up their own chemoattractant and they migrate in response to this. But actually, we had some evidence that many other immune cell types cannot self-generate gradients. They can sense gradients, but they cannot self-generate it. And so we were asking whether first of all, whether cells can surf on gradients, whether some cell type can surf on gradients generated by other cell types. And can we also ask, we could also ask, are there optimal principles for co-migration? You know, if you start coupling different cell types to each other, are there optimal types, are there optimal ways of doing it? Are there optimal parameters? Are there optimal sort of, signs of the couplings to make sure that they migrate together? And the motivation for this is, again, these beautiful in vitro experiments where they've mixed up two cell types of dendritic cells in blue and T cells in pink. And I'm not gonna go make you go through the entire data. The key features of the data is that these dendritic cells, the blue cells, they can self-generate their own gradients. So if you would put them alone, they would make sort of a traveling wave. But these T cells in pink, if you put them alone, they do nothing. They just do essentially random Brownian diffusion. They're not able to migrate directionally at all. However, the cool thing is that when you put them together, they can actually invade in a directional manner. And even more surprisingly, the pink cells, the ones that couldn't do anything on their own, are actually more efficient. So they're actually at the edge, at the front of the wave. If I run you the movie again, you can see that it's sort of surprising. The pink cells that were doing nothing on their own are actually better than the blue cells. So the idea that we had is that, okay, this pink cell seems to surf on the wave of the blue, but if anything, they're more efficient than the blue, which is, at least to us, was not completely intuitive, right? That it's a bit of an honor. Yes.
Are they seeded in a way that they are separated and then they see each other, or it's a mixed population?
It's a mixed population, but we have a bunch of experiments. I mean, we have a bunch of experiments. It's not really that they physically interact, essentially. So it's not that the pink cells are pushed by the blue cells, for instance. And the key example of this is that they have a mutant for the pink cells where they cannot sense. So the pink cells cannot eat their own chemogelatin, but they can sense it, basically. But then you can also make a mutant where the pink cells don't do anything, so they cannot actively migrate at all. And then what they do is they're completely stuck. So the blue cells move, and the pink cells are completely decoupled. So it's not mechanical. It's not that they're dragged or under friction or push. It's really active. It's really an active coupling through a diffusible field. Okay. So can we explain this kind of? Can we explain this kind of ideas, and can we use this kind of theories to explain these things? So what I'm gonna, the kind of theories I'm gonna write actually extremely familiar to, they're actually exactly, so what Mehmet wrote is essentially almost exactly what I wrote before. It's just a multi-component case. So essentially what we write is we write multiple cell types, right? So we have different, we can write an arbitrary number of cell types with density rho. And so these density rho's, right, they have some conservation equation where they diffuse at possibly different diffusion rates. And then all of them have an advection term and migrate up a relative gradients. So here you can see that this is the gradient A of your chemical field. And then the reason why the interaction is non-symmetric is that if you look at now the equation for the diffusible field A, field diffuses, but it's eaten up only by one of the cell types. So that's what creates the asymmetry in the problem. Of course, it's also that all of the cells can have different coefficients, right? It has different diffusion coefficients and different chemotactic coefficients, but it's also that only one of them can degrade the gradient. And then you can ask, okay, what sort of dynamics, what sort of collective dynamics do you get if you consider very generically these kind of things? And what Mehmet showed essentially is that you can, if you have two cell types, one that consumes and senses and one that senses only, essentially you can reduce the whole phase diagram to essentially two rescale parameters. The first rescale parameter is exactly the one I've mentioned before. It's essentially so here C denotes the consumer, so it's the cells that can eat and sense. And there's one parameter which is its advection, its chemotactic index, chemotactic strength over its diffusion. And the reason why this is important is that the traveling wave solution is controlled by the consumer, right? The consumer is the one that eats up the chemokine, so it's the one that that is key to generate sort of the whole dynamics. And so this is really important because of this, right? Because essentially, if this ratio is less than 1, I'm not going to have traveling wave solutions, and so I'm not going to have things that are very good. So if you look at so essentially these are the front, these are these are color coded by the front velocity of each of the cell types. If you look at the y-axis here, this is Kc over dc. Essentially, if this is less than 1, the dynamics is a bit boring. It's essentially diffusive. So all of the population is diffuse because even the consumer cannot, is not strong enough to make a chemotactic front. So this is one thing. But then the other interesting thing is the relative chemotactic strength of the sensors and the consumers. And what happens essentially is that you also have a pretty strong cliff. You also have a very strong sort of phase transition when this is 1. And let me just show you maybe actual profiles. So these are actually concentration profiles of the consumers in blue and the sensors. So again, this is like if their chemotactic strength is too weak, all of them too weak. Essentially, you just have if you look at them in time, it's essentially a bit of a diffusion. It's essentially a bit of a diffusion equation. So here, all of their chemotactic strength is weak, so they just do diffusion. Here, the consumer is weak, so it doesn't move, it just diffuses. The sensor is strong, so for a while it can make a small, because this makes a gradient. So initially, this one has a kick, right? Because this one, the sensors here senses a gradient just here, and so it sort of makes a pulse and it sort of starts moving ballistically, sort of very fast. But then it stops because the blue is not moving, so it's sort of slightly boring dynamics. But then what happens? More interestingly is the top side, so 1 and 2 here, where now the consumers can make a chemotactic front, and now the question is, can the sensors keep up or not? And essentially what happens is that there's essentially a strong phase transition happening if the sensors are weaker than the consumers. If the sensors are even 1% weaker than the consumers, because they don't reshape the gradients, they fall infinitely far behind. What happens is that the consumers make their own traveling pulse, but then the sensors are left behind and they're gone forever. Even if they're 1% sort of weaker, they're always going to go in the back. Whereas if you go now, if the sensors are much stronger than the consumers, essentially what they're going to make is that they're always going to be ahead. They're going to form their own peak ahead of the front, right? Because, I mean, they cannot go infinitely far because their speed is bounded by the consumer. The consumer makes the gradient, and so you sense these sensors sort of bump into a wall in a way because they bump in a wall where at some point the gradient is steady. But essentially, they're going to form sort of a gradient, a peak ahead of the front. And so, because there's a very sharp transition where the sensors need to be at least stronger than the consumers, this actually explains why you need the sensors, you need the T cells to be either they're useless and they're in the back, or they're always in the front, right? Because if they wouldn't be in the front, they would be infinitely far behind, essentially. So it's a bit of a binary choice. So there's a very, very strong design principles that the sensors need to be stronger than the consumers, and because they're stronger than the consumers, they're going to form peaks ahead. So that's a very simple prediction of the model, and that's something that you can see in the data. And so you can do quite a lot of analytics on this. You can actually derive through things similar to what I've shown you before. You can derive analytics for the speed of propagation. You can derive analytically the shape of the front of the pulse, which are exponential profiles that depends on the speed of the wave and the diffusion. You can predict. You can show that each of the two species decays exponentially with different rates, and actually you can extract the relative diffusion coefficients from the relative rates, from the relative profiles of this. The cool thing with this is that actually we had quite a bit of experiments on this because we knew we have experiments where you have only single cell populations. We can track individual cells to get diffusion coefficients. So we have quite a lot of experiments that we know by FRAP, for instance, the diffusion coefficient of the chemokine. So we know the dynamics of the diffusible field. So actually we can sort of constrain quite a lot of parameters. And so these are essentially the best fit parameters where we've sort of constrained everything. And what we predict with this is essentially a state where the consumers behaves as a quasi traveling front, whereas the sensors behave as a traveling peak, which is a little bit ahead of the consumers, right? That's a prediction based on independent fits of this on these parameters. So you can do two things. You can say, okay, am I predicting correctly my dynamics? And you can sort of again, these are experiments, so you can average, you can do chemographs in 1D and you can average them. And you can see that actually, of course, it's a little bit noisy, but over 3 repeats, you can see that the consumers look quite a bit like a traveling pulse, whereas the sensors look quite a bit like a traveling peak, which is in front. You can say, okay, this is a little bit noisy, so it's a bit hard to say. What's cool about having a traveling wave is that you can get much better statistics by just syncing up, by going in a co-moving frame. So what you can do is both in the theory and in the data, you can go in a co-moving frame to see if you have a traveling wave. And essentially, it works quite well. So if you see it's not a perfect traveling wave in the model. So individual lines are like different time points, but you can see that they're pretty close to a traveling wave. And if you're doing the same treatment for the data, it works surprisingly well, actually, where if you average, you can see that there's a pretty clear front for the sensor, for the consumers, and a pretty clear peak from the sensors. And you can see that actually you can predict quite a few things. For starters, for instance, we extracted that the sensors have larger diffusion than the consumers, and so this exponential is about 3 times sort of broader than this exponential, which is what you see in the data if you plot it exponentially. You can see that the distance between that front and the peak is quite well reproduced by this. You can see that the global length scales are quite well reproduced. There's quite a few features of the data that you can explain with this super simple model. Yeah.
I meant that it almost looks like there is a cusp. Would you say this is just because of the noise that it looks like that, or is there something hidden maybe?
No, I think it's linked essentially to so essentially when you move in a co-moving frame in the experiments, what you do is essentially you synchronize everything up until on the peak of one of them, right? And so if you have a noisy signal and you synchronize, I mean, if you have a bin signal and you synchronize the peak, what I think is it just amplifies a little bit this essentially. Yeah, we could. It's true. We could do that. I mean, the other thing that we've done is that here we go in the co-moving frame of the red cells. We also tried to go in the co-moving frame of the blue cells, and then the red thing is a little bit smoother actually. So we tried different things essentially. That's a good idea. We could also confirm the model. So this is just one thing on which to say. This is just one thing to say that. This is a surprisingly simple theory that has quite few parameters that you can measure. You know, if you think about it, I mean, you can measure them either by FRAP or by tracking of the cells or something. So it's something, but it seems to do quite a nice job at sort of predicting qualitatively and even quantitatively quite a few features of this kind of immune cell migration and how reciprocal, how sort of particles can interact through different things through a diffusible field. But the last few slides I want to talk about is. There was something that interested us a little bit with this, is that if you look at this parameter regime, it was sort of funny where all of the parameters are sort of intermediate. You know, they're almost all close to unity and a bit larger than 1. And we just wondered if there were some design principles or some optimality principles, you know, going back to, you know, what's the best way. If you think about now immune cell migration, what do you want your immune system to do? And we talked quite a bit with sort of with biologists. And there's two things so there's a few things that are a little bit obvious. So one thing that you want to do is you want KC over DC, you want it to be larger than 1, otherwise you don't have traveling wave solutions. So you want KC over DC to be larger than 1, and you want your ratio of chemotactic strength, Ks over Kc, to be larger than 1, otherwise your sensors are lost. You know. But you could say, why are they not much larger than 1? Then I would have like a super chemotactic system, it would go super fast. Why don't I do that? Well, we talked to biologists, and so one of the key things about immune cells, the immune cell types, that it's not just that they want to migrate far, is you also need them to interact with each other. So for instance, D cells and T cells, it's really well known that, for instance, they interact physically, they need to, I mean, essentially they need to learn to recognize antigen, and so they exchange information. And so what you want in an immune response is you want two different immune cell types to talk to each other. And so you want them to be spatially colocalized. And then we thought, what kind of parameter regime do you want if you both want robust migration, so you want cells to all migrate towards a specific target, but you also want them to be colocalized spatially? And then you're going to see actually that if you think about it, you're going to see that all of these things have trade-offs. For instance, you might say, why do I have diffusion of my cells? What I want is to be chemotactic. If I have like zero random diffusion of cells, I'm going to make heavy side functions that are going to be super efficient. I'm going to have like, you know, very little dissipation, it's going to be great. Yes, but then your peaks are going to be essentially heavy-side functions, and they're going to be completely spatially segregated, right? Similarly, you could say my sensors, I don't want them to risk being infinitely lost in the back, so I want my sensors to be super, super efficient at migrating. Yes, but if your sensors are super efficient at migrating, they're going to form a peak very far away from the consumers, and they're, again, not going to spatially interact. So then we looked at every parameter, And essentially, everything goes in the reverse direction, where if you want to migrate very efficiently, you want your chemotaxis to be super efficient. But if your chemotaxis is super efficient, you form like very, very narrow sort of peaks, which basically don't have very little overlap. So what Mehmet did is essentially to summarize everything in a phase diagram where, where essentially the color code here, I mean, you can do it in several ways, but essentially the color code here is a mix of, where essentially we try to mix, we want cells to migrate efficiently, but we want them to co-localize. So we had some essentially co-localization index, which is essentially a convolution of the two densities, where essentially what you want is, you want cells to share spatial overlaps. And what was cool with this is that it's essentially the optimal way is essentially the blue region, and so it's the region of intermediate parameter space. And if you put the star here as the best fit parameters that are inferred, it's actually something that's pretty close to like the best parameter region, the best parameter region, if you want to be again both robustly migrating and co-localized. So of course it's very hard to prove, you know, optimality in biology, but we think it's sort of interesting in the sense that it shows that if you want to have different populations of cells co-migrating, there's some really interesting trade-offs that's going to show up, and maybe sort of the data even sort of obeys these kind of trade-offs.
So
and that's it.
Another question that comes to my mind by that is, this could be also used as a mechanism to tune very quickly your response, because you may have this optimal parameter region, but you want to drift away very quickly when you need it. This is something that you thought about, you studied, you know, the variability of the position of that star in time. So the system can change the position of the star.
Yeah, the parameters can probably change a bit. I mean, it's a good question, actually. I mean, I think there's some. I mean, the thing is that the problem is that we don't know a lot of parameters in vivo, right? What's relevant in vivo? So, for instance, I don't even know. I mean, it's not extremely clear even what produces the chemoattractant in vivo. I mean, there's some idea that the endothelial cells produce it, so you make essentially highways of things that are, you know, you've made highways in your body. There's very little things we know. So that's a bit the issue, is that I don't know, for instance, I don't know what I mean, I'm not even sure what activates the immune response. I mean, is it sort of, what I would think probably is that you activate your chemotactic sensibility. I guess you could also make the chemoattractant. I'm actually, I need to read more about immune cells. I actually don't know. I actually don't know if what changes the most is on the size of the cells or on the size of the chemoattractant field. You know, I'm not so sure. We've been trying, there's one super cool experiment that they've done, which is that they've injected, you know, what they've done is they made a wound, and then they injected a bunch of fluorescent immune cells on one side of the mouse. It's an actual in vivo experiment. And then you inject sort of 10 cells, 100, 1,000, you know, you inject sort of increments of cell numbers, and you ask how many make it to the wound site. And essentially the fraction of cells that make it to the wound site increases with the number of cells. So, you know, if you inject 10 cells, essentially almost none of them make it. But if you inject 1,000 cells, 10% make it, which is exactly consistent with the collective dynamics, where you need some type of quorum sensing, where you need some type of, you know, collective behavior, you know, and to shape a gradient. Can we extract some parameters out of that in vivo? You know, maybe just this is. I need to read more on immune cells. Basically, that's my. I'm basically done. So I had another part, but it's way too ambitious. So if you have any questions, otherwise, this is basically a conclusion. Yeah.